Kwangmoon Park from University of Pennsylvania.
Thank you for the opportunity to present my work. Today I'm going to talk about learning causal network from Perturb-seq data set, including instrumental variable. So this work is a joint work with my postdoc mentor, Hongjae Lee, at the University of Pennsylvania. So what I'm going to address today, the main problem is about learning causal regulatory relationship among genes from perturbational single-cell genomics data. So this project was mainly driven from a particular data set called as Perturb-seq data, which is like an interventional version of single-cell RNA-seq data. But before that, I want to provide you some backgrounds for this type of data and the general single-cell genomics data. So for the last several decades, biologists and geneticists have put a huge amount of effort to collect diverse type of molecular features from human tissues in order to enhance the understanding on human biology. And the biotechnological advancement have led to the point where we can collect the high-dimensional molecular feature from each individual cell. And one type of, so that technology called as single-cell sequencing technology, and that was kind of highlighted as method of the year by Nature Method in 2013. And one type of such technology provides something called as single-cell RNA-seq. I think this might be a little bit more familiar to you than other type of data sets. So essentially, this is like an observational version of the data that I'm going to analyze for this talk. So from this sequencing technology, what you would get is for each individual cell, you get a p-dimensional numeric vector across genes. So if there are p number of genes, then it's going to be p-dimensional vector filled with the expression level of each individual gene. Each entry is represented by mRNA molecular count that this gene has transcribed. And you can think of each cell as a sample, and you would have IID samples across different number of cells. Then this is going to give you a nice matrix, which is cell by gene, filled with the expression level of each particular gene and a cell. Okay, so this is the observational version of the data set that I'm going to analyze. So the motivating data is called as Perturb-seq. So Perturb-seq is an interventional version of this single-cell RNA-seq. So this is also going to give you something similar to this expression level matrix. So the Perturb-seq, you get the cell by gene matrix, which is filled with expression. But here, what you can do is you can intervene on each individual gene's expression level by the lab experiment. So that is going to give you this matrix, but we have an additional contextual information on which gene was intervened for each cell. So that information is given in the data. So for example, this color that I colored over here represents which gene was actually intervened for that particular cell. So for example, for those yellow cells, the first two cells are colored as yellow. You can actually just know that for these two cells, yes.
Do you know it, or do you estimate it based on the gene expression?
So there are different type of technologies. So so gene expression is different thing. So you don't estimate it from gene expression. So there's a way to detect the guide. So I'm gonna talk more about the details how it's intervened, but you just know it. I mean, so there's a way to detect it. Yes, okay. Yeah. Not kind of infer from the expression level. Yeah. Okay. So I'm not sure. Yeah. Then for each cell, you will be able to know which gene was intervened. And if you just kind of condense this information into this binary indicator matrix, then each row, if you take a look at this, the entry that is filled with 1 denotes that this particular entry, the gene was intervened for each individual cell that corresponds to each row. Then there are a group of cells where intervention has happened, and there also is a group of cells where no intervention was happened. Okay, so how does the gene get intervened? So for each individual cell, you have a long strand called as DNA, and the particular functional region called as gene expresses the mRNA molecule. Then the way we intervene is we induce this dCas9 protein that is combined with this guide RNA. So essentially what this does is, let's say we have a target gene of interest over here. Then this guide RNA kind of detects which region to intervene on. And this dCas9, this protein, kind of does something on the expression level. So there are different types of intervention that you can do. You can either repress the expression level or activate the expression level, which means just boost up, or you can even knock down or knock out. But the motivating data is mainly about CRISPR-I, which means we try to intervene on one gene, then the expression level is going to be less than usual. Okay, so this is how the intervention is happening. And I mean, it is possible that you can intervene on multiple, yes?
A lot of acronyms, so can you tell us what's different between dCas9 and sgRNA?
So this is referred to as single guide RNA.
So this is just locates the...
This detects which region you want to perturb or intervene. And this is like a name of protein that does actually does repression or activation of the expression level. Yeah, I forgot the acronym for this guy, the actual term for this guy, but that's the protein that does actual repression or activation of. So this you can think of as like a thing that kind of finds the region that you want to, yeah, then this kind of binds together with this guy, and then the intervention happens. Yes?
So the repression and the activate is people use the same protein or different?
Different protein. For repression, you use dCas9 crab, and the CRISPR-A, you use different thing. Yeah. Okay, yes?
And you get it in with a virus.
Oh, yes, right. That's right.
Sorry, I'm trying to figure.
Yes. Yeah, so this just finds the region, and this is the thing that actually kind of knock down or knock out. Yeah. But yeah.
At least the way I like to look at it is a non-biology. A single guide RNA is like the GPS in your car.
Yes.
And then the dCas9 is the car that gets you there. And so it's
like a navigator.
So dCas9 is the virus. Then.
No, dCas9 is the protein that unwinds that DNA, and then single guide RNA is the word that tells you where to go.
Okay, and the virus is the one that gets into the cell.
Yeah. That's my understanding.
I mean, essentially that is. So technologically, it is possible that you can induce multiple guides and target multiple genes. But for since the motivating data only has a case where one gene was intervened for each cell, I'm going to consider only that case. And also, I'm going to consider the case where all the genes that we have observed are actually at least once intervened on multiple number of cells. Okay, so the data you will see is the expression level matrix and this perturbation indicator that tells you the design of the intervention. And you could possibly also have cell-specific metadata, which we can use as a covariate. Okay. So in principle, causal gene network can be learned, not just associations. What I mean by that is the causal gene network, which I denote as CGN, is stacked over gene expressions.
Okay, sorry, can you go back to the previous slide where you described this perturbation indicator? So this is, I guess there's some assumption that the perturbation, because you said you can learn the network, you're assuming the perturbations are randomized in some way, because there are multiple perturbations that are.
Yes, there could be some covariates, observed covariate technical factors that can influence this binding indicator. It's not, I mean, it could be fully randomized if the experiment is, well experimented. But there could be some covariates that can influence this binary indicator.
So it's possible, usually I thought the technologies, you get a library of interventions and you kind of...
Yes, I mean, we want it to be randomized, but...
Some virus is randomly infected.
Yeah.
But you're saying because of some technical issues, it may be that certain cells are more easily perturbed.
Yes, that could happen. Yes, that we adjust for in our procedure. Yeah. So essentially, in principle, we can learn the DAG over genes. To kind of showcase how this happens, I just wanted to give you a very simple illustrative example where we only have 4 genes, And each node corresponds to the expression level of a particular gene. And let's say this is the underlying DAG. Then say we don't have any idea on the network. And we can, let's say, intervene on each gene, each of the 4 genes. Then for a particular group of cells, let's say we intervene on a particular gene, let's say gene 1, to reduce the expression level. Then let's say we saw the expression level changes for the other genes. Then this will be a good evidence that the other genes, gene 2, 3, and 4, they are the downstream of this gene 1. On the other hand, if you intervene on just gene 2, and then reduce the expression level, and so that this guy changed, but the other 2 didn't change, then that would be a good evidence that this guy is a downstream or the descendant of this gene 2. So in this manner, you can kind of get the ancestral or descendant information for each individual gene. Okay? Then if you know, let's say for gene 4, you know that gene 1, 2, 3 are in ancestors, then what you want to do is just like conditional independence test to find out what are the direct parents. That's the insight. So with this insight, we can learn ancestor, descendant, or upstream or downstream, and then can learn direct causal links among the genes. So typical goal for this type of data is to learn a DAG over genes where H corresponds to the case where gene I regulates gene J. But here, the main challenge that I'm going to address is that the Perturb-seq comes with substantial unmeasured confounders. So there are actually several aspects related to this confounding. So biologically speaking, there are a lot of latent biological factors that can influence the relationship among genes. This could be like cell cycle, chromatin state, so forth. And also economically speaking, in a cost-constrained experiment, only key genes that the experimenters are interested in might be intervened. I mean, it is not the case that all genes are intervened from a data, or even they might observe a certain set of genes instead of looking at all of them. So this could potentially give us some omitted genes in the analysis. So if you kind of translate this into statistical challenge that's relevant to an observed confounder problem, and the perturbation indicator that I mentioned can be used as something like an instrumental variable. Okay. Yes?
For these latent factors,
Yeah.
some of them can be observed, or because of this cost constraint, that people can like cell type.
Yes, cell type, that's also kind of inferred from the data. I mean, cell type could be given as label, but these are not just given from the data. Somehow the biologists or the people who generate the data must infer what are the cell types. Yeah, these are kind of hidden, so they want to learn what that is. Yeah. Yes.
Can you go back to the previous slide? Sorry.
Yes.
Where you. So I've got it's here. There's a couple of questions.
Yeah.
One is, why does it have to be a cyclic?
Oh,
could it be a the actual network has cycles?
Yeah, I mean, in biology, it could be the case that there could be cycles, but we are just putting a model in order to kind of interpret it based on conditional independency.
Right, and I guess that leads to my second question, which is, I think the way you described this was very kind of simple, that you just kind of try to find descendants, like upstream, downstream variables, but you don't try to do more sophisticated analysis. Like these people who are doing causal discoveries, right? You look at conditional independencies.
This is more like a local approach. It's not like globally learning the DAG at once, but yeah.
So it sort of requires you to kind of intervene on every node.
Right.
If this is all you do is looking at who is downstream and upstream.
Right.
So it's quite inefficient.
I mean, I did some computational comparison with the existing method that also requires all the genes to be intervened, which is kind of global approach. Compared to that, the computational power was not there.
But have people not tried to use causal discovery methods? Maybe
Just like, yes, there's lots of people who have used causal discovery methods in this area. But to your first question, there's also lots of work on the optimal number of interventions you need to do to learn the graph.
Right.
Like Caroline Mueller and Frederick Eberhard and others have all these results going back like 20 years now that say like how many, depending on what assumptions you make about the structure, how many interventions do you have to do to learn the entire graph and stuff like that. So there's a lot of work on that question.
Okay, so it seems there's a gap that hasn't been translated here to this type of data. Maybe
I think it has.
it has.
This is more like a local approach than a global. You have a question? Okay. Yeah. So since this kind of confounding exists in the data, the main property that I care about is the robustness. I mean, you can think about many properties for your methodologies, but the property that I mainly care for my method is to have that robustness against omitted confounding nodes. So for example, this is two of the DAGs learned from a baseline method, one of the baseline methods that is developed for this particular data set. The one on the left is the DAG learned from the full nodes, and the one on the right is the DAG learned from the case where we omitted these yellow nodes. So you can see that two DAGs are just pretty different just by omitting this Y7 and Y8 from the analysis. And you can even see that the edge-specific coefficient changes dramatically just by removing these two genes or two nodes from the analysis. So this is the thing that we don't want. What we want is something like this. So even if we omit some of the nodes from the analysis, we want that the sub-DAG is well recovered, even if we miss some of the confoundings. Yes?
Sorry, but if you lose a local method, you say if I intervene on this gene, this expression has changed.
Yeah.
That doesn't require you to have no confounder because you've actually done the intervention.
So the confounding that I'm caring about is the confounding among the gene expressions.
Right, but if you say I intervene on X1 and X2 changed, that means there is a causal effect.
That can, I'm going to talk about it in a later slide, but for the descendant search, there's no worry on major confounding.
Okay.
But for the direct parent search, then we need to kind of incorporate some of the...
These methods don't just do this local approach you described, they do something else, and that's...
As far as I know...
vulnerable to...
Yeah, most of them were global approach. Yeah.
So people also have some kind of observational data where no gene is
Intervention.
Intervention. Okay. So basically, they have two data sets. One is this observation, the other is this intervention, and they know which, but different interventional sample can have different intervention variable, but okay.
Yeah, there are methodologies that tries to recover the DAG even with the, no intervention and multiple environments, but that's another method that could be applied here. But we're
So just so you're not directly intervening on the expression, right? So it's the intervention of the Perturb-seq, it's the introducing the virus. It's like an intention to treat kind of.
Yeah.
It is the IB.
Yeah.
And the expression itself.
Yeah.
So if you're trying to make a network between expression gene 1 and gene 2 based on expression, those could be confounded. Yes.
Yeah. Yes.
Okay.
Yeah. I have more detailed models behind it. So the binary indicator is some different variable, and that can influence the expression level of the targeted gene. And then we are more caring about the interaction between this gene and the other gene, but using the intervention on this. So we cannot directly set the expression level to the level that we want, but we just have a guide RNA that can do the intervention. Yes. So we cannot do dual operation on expression levels. Yes. Okay. So compared to this guy, what we want is something like this, where even if we remove some of the confounding nodes, the subclass to be the same. And you can here also see that the coefficients kind of are stable even before and after the omission of the gene. I mean, before even talking about unbiasedness or consistency, I wanted this kind of property to exist in the data method. Okay, so some more details on the data set. So for each individual cell I, you have a vector of expression level, which I call as yi. And there's di, which tells you which gene was intervened on. So this is, if you sum over this vector, that's maximum 1, meaning that we only intervene on one gene at max. And if this is entirely 0, that means that cell is control cell. Okay. And you could also have some covariates such as batch depth. These are observed things, but the main thing over here is that there could be unobserved vectors, which could be cell chromatin state, cell type, or even some potential missing confounding genes. Okay. So before I dive into the method, I just want to tell you the model that I rely on. So the model that we rely on is essentially an extension of classical measurement model for single-cell RNA-seq data, and we kind of extend that to the case we have a DAG over genes. So essentially, the Yij, which is a UMI count of particular gene at a single cell, that's coming from this Poisson process where this Li corresponds to the library size, and this lambda is like called as a latent expression level. So this is actually widely adopted by the literature. And with some proper priors on this lambda, I mean, if this is just like point mass, then this is going to be again Poisson. And if this is gamma, then this is going to be a negative binomial. And people typically call this mu ij as the true expression level, and the Yij as the noisy observation of this mu ij. So the key over here is that our model puts a DAG over this mu ij instead of this Yij, which is noisy. So it's kind of similar to causal representation learning in some sense. So for this log mu, which is the true expression level, we have a structure equation model. And this theta jk tells you something about the causal effect of this gene k on gene j. So that's the key parameter that we want to learn. And here is the dj, which is the binary indicator. This tau j represents how strong the intervention was for that particular target gene. And this x is just the observed covariate, and this ui is a unmeasured confounding, and gamma denotes the strength of the unmeasured confounding on this particular gene. So this is the structure equation model, and you also have a noise term that is required to be mean 0. So to tell you more details about confounding, I mean, the established literature talks about potential confounding of this D and expression level by the technical factors. So we kind of also rely on that. These can be all observed, so you can kind of regress them out in the method. But we also assume additionally that there's some hidden confounders that can influence the expression levels and potentially this edge that we want to learn over here. Okay, so the difficulty here is that the mu are not directly observed. These are true expression level, like that gives the data set that we observe, Y. And another difficulty is that this is confounding these two arrows, but the remedies for this, I mean, to give you a spoiler, we try to build a valid proxy for this mu. And as you can see over here, this D can be used as an instrument for learning this causal flow over here.
A couple of questions.
Yes.
One is, I thought, I mean, from a long time ago, I thought there was doing some zero inflated models, because in these single cell data, they often have a lot of zeros.
Yes. Yeah.
Is that you're going to ignore that?
Yeah, so there's actually, that's a very good question. So single-cell RNA-seq is very sparse data. Single-cell RNA-seq data are pretty sparse. So people have been putting like like the zero inflated Poisson model and so forth. And there's a paper by Rafael Izari and Harvard Biostat that says that the zero inflated model is not really the thing that you want. Okay. So they also rely on this. They also argue that this model is more appropriate for that type of data set. And the additional variability, maybe use some priors on there, add some more variability in there. That's more appropriate. That's what that paper mentioned, and also this paper talks about. I mean, but if you want a zero inflated model, then you can kind of put a different prior on this lambda. But that's not the thing that we rely on for this work. But yeah, zero inflation actually happens by having some normalization, log normalization +1, and the pseudo count kind of makes the zero inflation. That's the argument that Rafael's paper talked about.
Okay, I guess I'm not up to date.
Yeah.
And I guess the second question is, because you assume the linear model.
Yes.
I guess there are a lot of you're going to rely quite heavily on the linear model.
On the latent level, yes. Yeah. Right. So yeah, the remedies are using proxy for this log mu and these DJ as an instrument. So this is like a graphical illustration of the model that we rely on. We have observation on Y and D. And this mu is the true expression level where the DAG is applied. And we want to learn these red arrows. And also this theta jk, which is the causal effect of mu k on mu j. Okay. So based on this model, we developed a method called argan, where the goal is to recover DAG, the red arrows, and the data, despite the existence of unmeasured confounding U. So the strategy is to adapt the classical causal inference tool, including IV, to the CRISPR perturbation data to achieve that goal. So the main structure of argan is as follows. We first learn the ancestors and descendants for each gene. Then once you know the ancestor, we want to know what are the actual parent of that particular gene. So let's say this is the gene, then you know what are the ancestors. Then among the ancestors, you want to know which one actually is the direct parent. So this is the structure that this argan method has. And so let's first talk about ancestor learning. Okay, and for each of the component, I have identifiability result. And we also talk about how we can actually estimate those ancestors or their parents. Okay, so ancestors and descendants, how do we identify this? So under some classical assumptions, like as I think you mentioned this at the beginning, under some faithfulness assumption or like influentiality assumption, I mean, if this gene K is a descendant of this gene J, then already this DJ should change this YK, right? And also, if this YL was an ancestor gene of this gene K, then the intervention on this guy shouldn't change the expression of this guy because this is a collider. And this is under classical assumption like faithfulness as influentiality, and that holds even if you have this U. Okay, so in our paper, we provide identifiability result based on some weaker assumption. Actually, that's employing the notion of exclusive directed path. So the assumption that we rely on is essentially this tau j being non-zero, which means the perturbation effect on this target gene is non-zero. We need that, which is meaning that we want this to be strong enough. And the theta jk that builds a DAG, they should be all non-zero. Okay. So the informal theorem that we derived is the descendant and ancestor are recoverable based on this assumption. So the main idea is that, so without faithfulness or influentiality assumption, so we can miss some of the downstream genes even if you have an intervention on this guy by cancellation. But in Perturb-seq, the data set that we rely on, we have, even if this one node is missed from the test, we can actually have a gene that has an intervention in the middle. So we can kind of collect like all the descendants of descendants to recover the actual descendant, even if they are missed. Okay. So the ancestral information can be learned, and this using some Kant's algorithm, you can also learn the causal ordering. So that's the identifiability result, and how do you estimate that? So essentially, we know that gene K is not affected by gene J if this conditional independency holds. That's the null hypothesis. And based on our model, it's kind of equivalent to investigating the average expression level of gene K, whether the expression level of gene K changes per perturbation on gene J. So that's the equivalent null hypothesis. And that's actually same as saying that tau jk is equal to 0, where this tau jk denotes the regression coefficient for this dj, where we regress this yk on dj. Okay, this can be derived from our model that we have. So essentially, we want to test if this is 0 or not for each j and k to learn the descendants and ancestors. Okay, essentially, for each j and k, we feed the GLM using the cells where j's gene is intervened or controlled, then get the tau jk. Then, since this mean function is already so characterized here, under this null hypothesis that this tau jk is equal to 0, we can actually have a sandwich estimator for this variance, and then this goes to a normal distribution under the null. And the p-value also reaches to uniform as the cell number goes to infinity. So basically, we can collect all the p, j, k across j and k, and then do some type of like adjustment of p-value. And if this gene k is enoughly change it by the perturbation of gene k, then we can call them as the ones that are descendants of this gene j. Okay. And the ancestor can be also similarly obtained. I mean, you can call the genes k that contains this gene j as a descendant as ancestors.
Yes, I didn't get why you need a sandwich estimator. Are you just not doing a maximum likelihood?
No, because condition on this may not be Poisson or negative binomial. So you have a characterization on the correct characterization on the mean function, but this might not be just negative binomial or Poisson. It might be a mixture of them. So if you take a look at the original model, it has additional terms. Yeah, so it's not exactly Poisson or negative binomial. So that's why we need some sandwich estimator there.
Because of the confounders.
Yes, yeah. Okay. So this is how we learn ancestors and descendants. And given the ancestor for each gene, we want to learn what are the actual direct parents. Okay. So if you knew the ancestor for a particular gene, what you would want to do is just do a conditional independence test to learn the parents. But the thing is, this log mu is not directly observed. This is hidden. And also kind of confounded by this u. Right? So the strategy is to build a good proxy for this log mu, which has a protection against measure confounding and kind of represents these properties of this log mu. And we use this dik, the d as like an instrument variable for a two-stage QMLE framework. Okay, so one tempting approach that you want to do is maybe just do some instrumenting on this log mu. But the thing is that, I mean, with this, you might want to do this type of regression where you have this value over here, and this theta jk is going to be the parameter that you want to estimate. But this is not directly quantifiable from the data because log mu is not a thing that you have as a data. So instead, this regression, we cannot do it directly. So instead of that, we thought about this eta, which has this relationship, which is actually estimable from the data, and which has a relationship with this rk, with this alpha x. And you can actually approximate this guy with linear function with some mild conditions. So this eta is essentially the kind of like a proxy for this rk, or a surrogate for this rk that we can use for regression. So this regression is infeasible, but you can do this regression by using this equation over here, and you still have the same theta jk over here. So this you can estimate, so you can do this type of regression using the proxy variables. Okay, and then also if this mean function is correctly specified, then you can also estimate this theta. And if this theta jk is non-zero, then we call that as a parent. So that's how we can identify this. So how do we actually estimate the parents if we can identify them? So essentially, for all the ancestor nodes of the particular gene J, we estimate or predict this eta k using this regression, where we regress yk on this dx and L. That's the first stage regression, just like the two-stage regression framework in the IB. And you do that for each of the ancestor node. And then you have this proxy variable ready. Then you can do the second stage regression to learn this theta jk. And since this is random, like this is a predicted value, we need to account for the randomness of this guy. And there's actually a literature that talks about how to adjust for the variance for this guy when this is obtained from another optimization problem. And with this variance, you can actually reach to a normal distribution under the null hypothesis that this is 0. And you can also obtain the p-value. So I want to talk about, so since this is kind of similar to like two-stage regression, I just wanted to tell you that the D that we use are kind of valid as instrument. So if you take a look at this eta over here, you're actually just using this dk and this ancestor of dk, or ancestor of this guy's dk, to do the first stage regression. then we only need to consider the IV assumptions holding for these two guys when we want to learn the H over here. So you can see that this U and this D are independent, conditional on this X. This is met, and also there is no direct link from this D to this guy over here. So that's the second assumption. And third is that this should have a direct effect on this gene that we intervene on. That's already assumed in our method. So these all validity assumptions for the valid instrument are met in this regression. So that's how we estimate the parents. But even if we have a parent set for each individual gene, simply by concatenating them, we cannot get. You're not guaranteed to have a cyclic DAG, right? If there's a sampling error, that could be the case that we have a cycle. So we develop an algorithm that is guaranteed to give a DAG, where the inputs are the descendant set that you obtain or ancestor set that you obtain, and the FDR cutoff that you want. So to give you just like a heuristic or idea of this, so among the nodes existing, we find a node with the minimum number of descendants and then put them at the bottom, put that one at the bottom. And then we already know what are the ancestors. So we estimate this eta for those of the ancestor of the bottom node. Then we can just fit the second stage regression and then get the p-values, right? Then we do some sort of p-value adjustment to learn the actual direct parents. So after this stage, removing this guy, among the other nodes, we find the one that has the least number of descendants. And we go through this procedure to build a DAG from bottom to the top. And for this adjustment on this p-value, we need an all-line FDR because it's a sequential approach. Okay. So we did a setup simulation. So we built a DAG with different sizes of genes with unmeasured confounding and CRISPR interventions. In our paper, we did two types of study. One was robustness to like omission of genes, and the other was DAG recovery with latent vectors. But for this talk, I'm going to talk about only this one. We compare our method against this method called S-INSPRI, which is like a global approach applied to Perturb-seq data. And we also compare argand against this naive method where you use this proxy. So the ancestor and descendant information is shared. I mean, the same for this guy, but this is using this proxy, which has no protection against unmeasured confounding. Okay, so this is the true DAG, underlying DAG. And this is the DAG that we recover even when we remove this Y7 and Y8 from the analysis. And you can see that when these two nodes are removed, you can see that INSPRI and naive are having kind of, yes. Okay. Yeah, they're suffering from recovering the DAG. And you can see here that even before talking about consistency, the coefficient changes dramatically just by removing the edges, nodes. And this blue line denotes the true value, 0.5 and negative 0.5. You can see that these are kind of clustered around the true values. Okay. So motivating example. So there's a data set from 2022, Perturb-seq experiment on K562 cells. It's a large panel data which contains like 2,000 genes, where each gene has some essential functional role. So for the analysis, we use the perturbation with sufficient number of cells, let's say 100 cells for that perturbation. And we also require that the perturbation effect is strong enough because that's the assumption that we also have. And we also adjust for the covariates such as batch or mitochondrial percentage. And we did two sets of analysis, one for biological evaluation, the other for more like exploration. So this is more like with smaller DAGs, and this is with the larger DAG. So I want to show you the sanity check result. So we wanted to check if the H coefficient data that we learn is actually causal. So in CRISPR-I, what we intend to see is that the targeted genes expression level decrease, right? That's the interpretation that that's the explanation that I had in the beginning. Then if the causal effect between this gene K and the child node gene J is positive, then the intervention effect of this gene K perturbation on this mu J should be negative, right? Because we are just multiplying minus and plus sign. On the other hand, if this was negative, this data was negative, then this intervention effect of D on this mu should be positive, because this is minus multiplied by minus. So we wanted this type of sign flip to happen between the intervention effect and the causal effect that we find out. Okay, from the real data actually. So we for as an example, we picked one gene and collected all the parent nodes. Okay, here the edge color represent the sign, and the width corresponds to the coefficient magnitude. And we took a look at the expression level of this guy per the perturbation on this. And for example, if we intervene on this bars gene, you can see that the expression level decreases. The expression level of this child node decreases, which is meaning that the intervention effect is negative. Then you can immediately see that the coefficient, the theta jk, is positive. And actually, you can see this type of relationship for all the parent nodes for that particular gene. We did an interesting experiment over here. We reversed the child and parent role over here to see if the expression level of the parent gene changes when we intervene on child gene. So basically, we took a look at the expression level of this VARS gene when we intervene on this MCM3. And you can see that the sign relationship does not appear over here. And you can also see that the change of expression when we perturb the child gene and is not really significant before and after intervention. Okay, so this is kind of showing us that the edge that we learned is not just associational, but kind of causal. We did this globally, so we collected all the, on the y-axis, we collected the intervention effect of this gene K on gene J. And we also multiplied that by the negative sign. And on the x-axis, we put the edge coefficient that we learned from the data. And here you can see that the 98% of the edges show such sign flip between the intervention effect and the theta. On the other hand, if we reverse the child and parent, then you can see that y-axis is pretty much flat. And reversing the child and parent role kind of shows about only like 55% sign match, which is kind of similar to random. So this is showing us that the argand's edge directions are causal, not just merely associational. So we did this type of experiment as well. We took a look at the DAG, learned from the case where we include this MAT12 gene and exclude this MAT12 gene from the analysis. And you can see that the remaining edge coefficients are pretty much the same, even if we omitted this guy from the analysis. And you can see that the coefficient didn't change that much, even before and after removing this single gene over here. But for this baseline approach, you can see that the coefficients changes significantly just by removing a single gene from the analysis. Okay, we did some biological evaluation. So there's a protein called the transcription factor. We collected them relevant to this the cell type that we analyzed. So the biological insight is that if this gene J regulates this gene K, it is likely that transcription factor, the protein, binds to both of the genes. So we kind of checked how many of these edges show enrichment of the transcription factor. Then turned out, so this is our this is our edges that are called as significant. And you can see that the enrichment is higher for this for our method compared to other baseline approaches. I talked about the larger scale analysis. We employed like 400 genes over here and drew a DAG. We did some module clustering of the DAG. And you can see that this result kind of recovers distinct functional modules, capturing some regulatory programs governing like core essential processes. So we also took a look at the in degree and out degree of this entire DAG. And the structure actually is similar to the network learned from external data set. And I mentioned that there's a protein called as transcription factor. This is so if this gene that generates transcription factor is existing in the DAG, it is likely that this gene that generates transcription factor has more child genes or descendant genes. Turned out like 95% of the transcribing genes had higher out degree than in degree, which is actually biologically making sense. Okay, so this is it for my talk. The summary of argand is that we can do robust causal discovery and inference in Perturb-seq data. And as I show you, it gives a trustworthy causal directionality. And we have seen also some support from external biological signals. So this is it for my talk, and thank you for listening.
Questions? Yeah.
Thanks for the very nice talk. I have a couple of questions. I guess the first one is maybe going back to what Chin-Yan mentioned. Like, so there is this work on causal discovery from purely observational, like gene expression. But then people have also tried to incorporate interventional information, maybe not so much with RNA, with Perturb-seq, but with like SNPs usually. And so people have combined like PC algorithm or other kind of like classic causal discovery algorithms with this interventional information. And I was wondering if, so in your case, what maybe was like closest in spirit to what you're doing is like combine the FCI algorithm with the indicators of what's intervened on. And I was wondering if you had any thoughts about that.
Yeah, actually, we initially wanted to start from that, like some invariance related, multiple environment related methodologies, but we kind of relied on this. If you recall, we relied on this model that is kind of extension of the classical RNA-seq model. So yeah, we kind of chose to rely on that and then kind of build our own method instead of doing some, employing some of the ideas from those type of methodologies. And also, it would be interesting to look at that existing methodologies performance in the data set and also simulations, but we didn't do that in our paper.
Yeah, I mean, I think it's consistent with with all your assumptions, basically.
Yes, right.
It's just you get a different object at the end, though, so it's like you have to be careful about how to compare, I guess.
Yeah.
But then I guess a follow. So yeah, I'd be happy to talk more about that offline. But also relatedly, can you say more about why you're assuming acyclicity here? Since like, so usually the reason people don't like to allow for cycles is because it's hard.
Mm-hmm.
But it's actually easy when things are linear, and like you're assuming a linearity. So like I think you could do everything you're doing and allow for cycles without changing very much.
I thought that like interpretation is much straightforward if you have a DAG. I mean, obviously there are cases where you could have some cycles. So if you take a look at here, even if you reverse the child and parent, we see some significant change. But yeah, that's the model that DAG is the model that we put because I thought that the interpretation is much straightforward.
But I think the interpretation is the same. I mean, the meaning of all the edges is the same thing. It's that when you perturb one thing, the other thing changes. But it just means that you could perturb A and B changes, but you can also perturb B and A changes. And it would change your procedure a little bit because now you could be both a descendant and an ancestor. You have more options for when you're doing your whole like figuring out who the descendants are part. Now you have kind of more options. But yeah. I think it's very consistent with the assumptions you have to extend that way. And I think if I remember correctly, you cite that inspire method. Yeah. They allow for cycles. They don't allow for confounders, I think, but they do allow for cycles.
Yes, they have some options to estimate it, with cycles or without cycles.
Yeah. This is not a criticism. I'm just wondering, like, as an extension of this, that could be a natural next step.
Sure. Yeah. But yeah.
Yeah, I should think more about that actually. Let's discuss.
Yes.
Thanks again for the nice talk. So I have a question about the log linear model that you had. So like, I guess I understand that you can't observe the mu I, j, but I thought there are procedures by the Sarker paper that gets the posterior mean of that mu. I'm getting this information secondhand.
I think that's similar to what we're trying to do, I guess. Yeah.
So, because one way, in addition to what you're doing in Proxy, is to just use the posterior mean, and then kind of go through this approach. So you're saying it's roughly similar to what you're currently.
Yeah, except that we're regressing out. We're doing instrumenting using the DIJ. Yeah, and typically people just use this they take logarithm of this y and divide by library size and add 1. And that's the point, actually, the paper by Rafa kind of.
I see. And maybe one more follow-up. So another way, I know the Sarkar paper did this, but another way is that you could put the technical noise as part of the Poisson measurement model instead of the latent scale or the latent expression as x's.
Oh, like the
Layer of yij instead of the.
Because I believe
yeah.
in your measurement model, I think that the y is related to mu, and then there's a mu is related to x's, but you could have y directly related to the measurement itself, or the counts are directly related to the technical noise x, and then mu is just a straight up, it doesn't rely on x's. But I don't know if you thought about this.
So you're saying having a node x over here and having a direct edge toward here. That's that. Yeah, that could.
I mean, it's a different assumption about the measurement, how measurement works.
Right,
I was
right.
curious.
Yeah.
We relied on this because we wanted to put everything in one linear equation, but that could be, yeah. But I don't know what's the intuition for, like, insight for putting that, having like a direct edge on.
I guess I was thinking more like batch effects. Like in some sense, the batch effect is not affecting the latent; it's affecting how we measure and calibrate 0, 1, 2. But that again, that all depends on what you think the batch effect correction is doing. Whether it's at the latent scale or at the observed measurement scale. But yeah, I know I think the Sarkar paper did it in the latent scale, but I was just curious your thoughts.
Yeah, I haven't thought about it.
We also followed down question about the cycle. So
Yes.
is this so is there a component like in your method like in deal with maybe the situation where your the estimated parent of J or ancestor is also include K, but the estimated parent K or ancestor also include J. So is there a parent in your method to address this? And this is due to you assuming a cyclicity or?
Yeah, so the algorithm that I have, since this is kind of building from terminal node and sink node to the root node, there isn't a gene that is like, yeah. But from our algorithm, but it is possible that one gene has the other one as descendant, and this one also has this guy as a descendant. So it could be the case, but the algorithm kind of puts the one with stronger descendant signal as the one at the bottom node, and then.
Okay. So if you do not assume a cyclicity, then you don't need this kind of.
Yes. So if you just I think that's relevant. So if you don't rely on this algorithm and then just basically learn the parents, then we can kind of concatenate all the parent set and then build a cyclic deck.
And also a quick question, like you have some FDR control in your method, but this is you have some FDR control.
Yes.
But this is just for reduce the discovery set instead of you are not really want to control FDR. You just want to reduce the discovery set.
Yes, we wanted to induce some level of sparsity in the deck, not just having everything that is significant.
So the goal is not to control that error.
Yes. Yeah. I mean, if you. Yeah, in this case, we had huge number of cells compared to the genes, so we thought that the distribution can be directly obtained. So we have p-values. We wanted to be a little bit more sparse, so that's why we relied on it. Yeah.
Coming back to the kind of global versus local approach, it's not very clear why you could not do a global approach here. Essentially, you have a linear FCM among these latent variables, your true expression.
Yeah.
Then you have a measurement model, which is a Poisson.
Yeah.
And in principle, you could have just kind of fitted this model.
Yeah, actually.
Together.
One of Hyunjung's students is working on a global approach. I think it's already archived.
A fully global approach is too sensitive to this particular model, but this feels like something could be done in the middle. Because what you have done is basically you do a local analysis, then you estimate it, some descendant sets, like qualitatively, then you try to kind of combine those qualitative sets. But that feels a little bit inefficient in the way that these things are combined.
Right. The issue that I saw so the ISPRI, that the method that I compared against is the global approach, but it's not relying on Poisson or something. But the issue that I have is that when they estimate something similar to my descendant set test statistic, and then they do some sort of inverse to learn the actual coefficient. Yeah. The problem is that the subset of inverse and inverse of subset is not the same. Like if there's a sampling error over there. So there, I think omission of some confounding nodes make problem, I think.
I think it's there at the population level there should be the same.
Yes.
But like at the estimation level, like I did notice.
But I felt there's something in the middle that you don't want to be completely global because it's too sensitive.
Yeah.
You don't want to be completely local because it's not using all the information. But maybe somewhere in the middle.
You can tweak INSPIRI a little bit to get something in the middle is what we worked on, but like it really depends on the signal to noise ratio on.
Yeah, like I also had this slide, I didn't notice that, but with a very small noise, you can make subset sub-DAG to be pretty different compared to the full one. Yeah, it's kind of insensitivity against that. Thank you.